Assessing possible, even subtle, health effects resulting from exposure to nanomaterials is challenging for several reasons, including the heterogeneity of particles leading to different effects and the lack of available tests with a no sensitivity and specificity to detect physiological and biological modification clearly related to nanoparticle exposure. These issues has been addressed during conference meeting and by working groups. So this area of research now is considered essential to carry out reliable field studies considering any possible health effects associated with exposure scenarios. Biological monitoring has been used for decades as a fundamental tool in occupational health risk assessment and occupational health practice. Biological monitoring consists of standardized protocols aiming to the periodic detection of early, reversible biological signs which are indicative, if compared with adequate reference values, of an actual or potential condition of exposure, effect of susceptibility possibly resulting in health damage or disease. Biomarkers are used as a surrogate indicators of designated events in a biological system due to the inaccessibility of target organs. It is thought that biomarkers are more directly related to the adverse effects which one attempts to prevent than any ambient measurement, and this supports the use of biological monitoring in risk assessment. On the other hand, biological monitoring is more complex in terms of standardization and interpretative effort as compared to ambient monitoring. Since relies on the use of biomarkers, a toxicological knowledge is needed for the interpretation, and ethical issues should be addressed as generally required in human studies. So, there are obviously several issues in the development of biomarkers for nanomaterials exposure. One key point to be carefully considered in exposure and health effect studies is the determination of the dose which can reach target organs and elicit biological responses. The internal dose for traditional chemicals is usually assessed by both the amount of the chemical or its metabolites and the product of interaction with target biomolecules, for instance DNA or proteins. So the development of appropriate biomarkers requires the knowledge of toxicokinetic and toxicodynamic data, which are still lacking for many classes of nanomaterials. Nanomaterials cannot undergo biotransformation. The particle dissolution may affect organ distribution. For some metal nanoparticles which release metal ions or can dissolve in biological media may be detectable not as particle but as metal species with appropriate analytical methods currently used. Whereas low-soluble biopersistent nanomaterials such as carbon nanotubes or titanium dioxide show as low clearance, leading to the tendency to accumulate over time with exposure. Although understanding target organ dose is problematic, obtaining quantitative data is essential for dosimetry model validation, build dose-response relationship, and ultimately for ascertain the range of exposure with no observed adverse effect. To estimate the dose retained at the portal of entry, there were put in place different methods. For instance, several toxic metals and trace elements are detectable in exhaled breath condensate, an aerosol representative of the lung environment, and this novel approach may represent a significant advance over the analysis of media, which are not as reliable, for instance, for interference due to complex matrix, and reflect systemic rather than target tissue level of element. By the brief analysis, tissue dose of pneumotoxic metal has been assessed in workers occupational exposed to different metals like cobalt or even chromium 6, hexavalent chromium. Although I think it needs further validation with different aerosols and model nanoparticles in selected exposure scenarios, pilot field studies have shown the suitability of such an approach to give estimate of the dose of particles deposited with the respiratory tract. Occupational exposure to welding fumes, ultrafine carbon black, or diesel exhaust have been associated to respiratory and cardiovascular diseases. Although not all nanoparticles cause inflammatory via mechanism involving oxidative stress, some manufacturing nanomaterials have clearly demonstrated to have pro-oxidative and pro-inflammatory properties. So it is important to assess effects at the portal of entry, at the main portal of entry in occupational setting. Quantification of lung inflammation is currently based on invasive or semi-invasive methods, and the measurement of inflammatory biomarkers in plasma and urine, which are likely to reflect systemic rather than lung inflammation itself. Brief analysis has been suggested as a promising non-invasive approach that allows the identification of the inflammatory and oxidative stress biomarkers involved in the pathogenesis of various respiratory conditions, both for clinical application and for investigating occupational lung diseases. Different biomarkers of effect can be determined in ABC, for instance, peroxidation products, leukotrienes, hydrogen peroxide, glutathione, and as well as various classes or volatile organic compound, a wide variety of carbonyl compound, which are generated as secondary oxidation products during their respiratory burst, in particular saturated aldehydes. Air pollution and occupational studies have produced an impressive body of biomarkers upon exposure to particles or specific components like, in this case, organic or elemental carbon from outdoor sources. The panel of available biomarker includes inflammatory endpoints, biomarker of platelet and endothelial activation, antioxidant capacity in blood. A number of nanomaterials can directly affect endothelial cell metabolism via oxidative stress generator and endothelial repair capacity, resulting, for instance, in changes in expression of vascular adhesion molecules and intercellular adhesion molecules. Interestingly, the above biomarkers have been determined in cross-sectional investigation involving some hundreds work in Taiwan, potentially exposed to nanomaterial, mostly titanium dioxide, the carbon nanotube, silica, and nanosilver. In spite of some limitation of exposure assessment, workers showed statistically significant changes across exposure risk classes for high-sensitive C-reactive protein levels and inflammatory cell activation markers, interleukin-6 and fibrinogen. Moreover, the depression of antioxidant enzymes like superoxide dismutase and glutathione peroxidase were associated with nanomaterial handling. I think that this panel of biomarkers can represent a good candidate for human biomonitoring following nanomaterial exposure. Panel studies and cross-sectional investigation of health effects of particulate matter exposure have shown consistent association between exposure to combustion-derived particles and products of oxidative damage and lipid peroxidation products, which can be considered as biomarkers of biologically effective dose. For instance, base excision repair of oxidative damage to DNA measured in urine, such as 8-hydroxydeoxyguanosine, seems to originate mostly from the oxidation of the deoxynucleotide pool and does not represent solely repairing excretion of the oxidized DNA guanine. T-bars and hydroxyguanosine have showed predictive value over lung cancer, for instance. Pilot studies suggest that, we know that now that also engineered nanoparticle, at least it in vitro system, but also some from some pilot study, can raise similar biological responses. Pilot studies suggest that several biomarkers of oxidative stress to lipids, nucleic acid, and proteins are persistently elevated not only in workers exposed to traditional dustose fibers, but also in workers exposed to nanomaterials independently for smoking habits, thus indicating that chronic exposure can maintain an inflammatory status detectable by oxidative stress biomarkers. Recent research also suggests that many nanomaterials can raise epigenetic mechanisms which have been implicated in different mechanisms of disease. And besides conventional genotoxicity tests, DNA methylation as well as other epigenetic biomarkers could reveal new mechanisms of action of nanomaterials. Since nanomaterials can potentially affect multiple cellular function and interfere with many components of the cell machinery, it is difficult to determine with conventional assays whether biochemical or functional changes can be material-specific or exposure-specific. This lack of specificity could affect the meaningfulness and hamper the applicability of biomonitoring to nano-objects. However, advances in the system biology and omic techniques can help to identify specific fingerprints of selected nanomaterials. For instance, comparative proteomic studies have shown strong similarities in the pulmonary response to different nanomaterials with well-known hazardous particles and fibers as measured by global proteomics. These have been applied for different materials as compared to traditional materials, for instance, single-wall carbon nanotubes and asbestos, or silica nanoparticles of different size and acute phase proteins. Moreover, a toxicogenomic approach has been used in identifying patterns of cellular perturbation in specific pathways through the identification and quantification of global shift in gene expression in cell models challenging with nanomaterials, or also in vivo studies. Pulmonary exposure to carbon nanotubes resulted in an elevated series of measurable potential biomarkers in blood, including gene expressed in the circulating blood cell and or soluble protein not unique to the type of particles. Interestingly, several genes were activated in the circulating blood cell, but not in the lung after pulmonary exposure to carbon nanotube. For instance, osteopontin, a marker of early mesothelioma appearance. In conclusion, biological monitoring is believed is thought a powerful tool, but its applicability to engineer nanomaterials requires the development of appropriate biomarkers of exposure and biological effect, considering the unusual properties of nanomaterials and based on a robust knowledge of mechanisms underlying such changes. Inflammation represents the main mechanism of injury for several nanoparticles, but specific physicochemical properties can trigger more complex pathophysiological events. While future studies should undoubtedly address the specificity of biomarkers, the priority now is not to assess disease endpoints, but to evaluate whether quantitative changes in already available biomarker, selected biomarker, can occur in groups of exposed workers. The sensitivity, that is the ability to avoid a false negative result, is fundamental for preventive purposes, whereas the specificity, that is the capacity to avoid false positive result, is usually more important for diagnostic purposes. Biomonitoring should also represent a valuable component of an integrated strategy and a proactive approach of risk assessment and management. This is not only an opportunity for companies committed with the responsible development of nanotechnology, but also an ethical obligation towards all workers' population that everything is done for the workers' safety and to detect any possible health threat related to nanomaterial exposure. Thank you very much for your attention. Any question?